Okay, it's saying recording has started, but that's what happened last week and I didn't get anything out of it. Okay, so I'm now going to share the screen. So just give me a few seconds that I'm going to share the screen with you. We will continue with our notes. And I just want to ask, are there any questions regarding the calculations activities? Are there any questions? I didn't get anything on the group, so I think it was clear. So if there's any questions, please feel free to send through your questions. Thumbs up if you can see the screen. I'm having RNA structure for the notes. So it's the notes, RNA structure. Please do thumbs up if you can see the screen on your side. Okay. Okay, I didn't see that, guys. I need to know that you can see my screen. Thumbs up if you can see my screen. I'm just seeing a few hands that are raised. So I'm not seeing the thumbs up. Okay, thumbs up if you can hear me. Okay, cool. Can hear me, but can you see the screen? I didn't get thumbs up for the screen. Okay, cool. All right. Okay. If there's anything on your side, please do unmute immediately and let me know because I'm not aware of what is happening once I project my screen. Okay, so we covered DNA and RNA in last week's intro lesson, and that was a very important part of our introduction because that's what our topic is about. Now, we covered a lot about DNA. We spoke about DNA being a double-stranded molecule. We spoke about DNA having a double helix shape. We spoke about the discovery of the DNA structure by those 4 scientists who were working in pairs. We then also looked at what we call a nucleotide, which is a monomer or a building block of a DNA and an RNA molecule. And we said a nucleotide is made up of a phosphate group, a sugar, and then a base. Then we said in DNA, there are 4 bases, A, T, C, G, adenine, thymine, guanine, cytosine, in no particular order. Then we said in an RNA strand, there are 4 bases. Now, the same bases would be A, C, G. But then in RNA, we do not have the thymine base. We have the uracil base, the U. So T is replaced by U in an RNA structure. I'm just recapping what we covered last week. Then we concluded the lesson by looking at the DNA calculations, which basically tell us that whatever number of adenine bases that we find in a section of DNA will be equal to the number of thymine bases that we find in that very same section. Same rule applies for C and G. I'm seeing messages on the group. I don't know what's going on. I'm trying to join. Okay. So that's what we have. So in this diagram here, or in this structure that you are able to see, we are basically having information about RNA. This was covered last week. So we'll have a phosphate group, a ribose sugar, that's the type of sugar that we find in RNA, and a nitrogenous base. Now, a base can either be A, G, C, or U. So I'm going to try and move very fast. Here, we are having the RNA structure, which is a single-stranded molecule, meaning it's only one strand, unlike a DNA, which is a double-stranded molecule. There's 3 types of RNA. I mentioned this. It's your messenger RNA, transfer RNA, ribosomal RNA. So for messenger RNA, you are allowed to just write mRNA, but your M should be in small letters and RNA should be in capital letters. That is important. If you do it the other way around or you mess it up, you won't get a mark. Now, this is a type of an RNA molecule that will copy a nucleotide code. Now, when we speak about a nucleotide code, it's nothing new. We are basically speaking about the sequence of bases. The sequence of bases in that DNA molecule being T, A, C, G, T, T, A, whatever sequence it is called a nucleotide code. Now, this type of RNA is going to copy this code from DNA and then deliver it to the ribosome, and that will take place during a process called protein synthesis, which I'm not sure if we'll be able to cover in this lesson or not. Time will determine that. Then we have another type of RNA, which is called transfer RNA. If you don't want to write the full name transfer RNA, you are allowed to just write tRNA. However, your T will need to be in small letters and RNA will need to be in capital letters. Now, this is a type of RNA that is going to transfer amino acids in the cytoplasm to the ribosome. When we speak about amino acids, I covered this, we are speaking about building blocks of proteins. So remember when we have nucleic acid, someone's mic is on, not sure if that's for a question or it's just on by mistake. Someone's mic is on, and now I need to go in here and try and mute everyone. Okay, so then this type of RNA, transfer RNA, it is going to also play a role. It might not make sense now, but I will explain it during protein synthesis. Then ribosomal RNA, if you don't want to write the word ribosomal, rRNA, but now your first R will need to be in small letters, and then the rest of the letters RNA will need to be in capital letters. So this one will form the structure of the ribosome, and this is where protein synthesis will take place. Now that's a very important section, but you will get to understand the function of each type of RNA when we do that process. Then when we speak about a strand, which is called messenger RNA. Now, this is obviously a single-stranded molecule. It's just one strand, as you can see it here. It's a single-stranded molecule. Now, when we look at the bases that we find in the strand, we read the bases in threes. So base 1, 2, and 3, then 1, 2, and 3. So how I would read them is GCU, then a little pause, ACG, a little pause, GAG, a little pause. Now, 3 bases in a messenger RNA strand, they are called a codon. So this terminology is very important. So 3 bases, 1, 2, 3. So this is codon 1 because 3 bases are called a codon. Now, we're having a list of bases in this strand, which is a messenger RNA strand. There's a lot of bases, but then we categorize them into codons, or we count them as codon. Codon 1 with these 3 bases, codon 2 with these 3 bases, 3, and so forth and so on. This is important because when we start speaking about transfer RNA, we're going to speak about anticodons. So the terminology here that is important, 3 bases in a messenger RNA strand. It's not in any strand. In a messenger RNA strand, we call them a codon. And then when we find 3 bases in a transfer RNA strand, we call them an anticodon. So in a messenger RNA strand, codon. In a transfer RNA strand, those 3 bases, we call them anticodons. Now, what we also need to know is that the anticodons are going to complement the codons. We will do that when we do protein synthesis. So if, for example, we're having UCA as our anticodon, this UCA will need to complement the messenger RNA codon. U will be complemented by A, and C will be complemented by G, and A will be complemented by U. So that means this AGU will then be part of our codons. My pen is also just acting up, but I think it's more important to listen to what I'm saying so that at least you can read this codon. This is supposed to be codon. Okay, I'm trying to rush, but the terminology is fine now. Then this, we are basically just getting differences between RNA and DNA. So when we look at a double helix molecule, that is for DNA, RNA is single-stranded, DNA contains nitrogenous base thymine, RNA will contain uracil, DNA contains deoxyribose sugar, RNA contains ribose sugar. DNA is known to be a very long molecule compared to RNA, which is known to be short. Now, I would say the first four that I've mentioned are very important. The last two, I think in the previous 2 years, they haven't really been entertaining them that much. Not that they are entirely incorrect, but they're not really emphasizing much about them. It's all about these points. So comparing that one is single-stranded, the other is double-stranded, the nitrogenous base, the type of sugar, and the length of the molecule. So please try and remember this table. They do ask it quite often, and they ask it differently. Sometimes they can say differentiate. In that case, you don't need to draw a table. But once the question says tabulate, you need to draw a table. And once they say tabulate, please draw a table and write the heading of that table. By just doing that, you get 1 mark before you even answer. So you tabulate and the heading, that's 1 mark before you even answer. Then, in terms of this question, I think we did this questions. If we didn't do them, I can gladly send a memo on the group so that at least we are able to just move past them because they are simple. Now, this is what the lesson is about. So this part is very important, very, very, very important. Now, we speak about DNA replication. This very same DNA that we've been speaking about, that we say we find in our cells, it's able to replicate. What we mean by that is DNA is able to make an exact copy of itself. Exactly that. It's able to make an exact copy of itself. It's able to make a replica of itself. Now, there's few things that you need to understand about this process. One, what is this process actually? It's about DNA making a copy of itself. Where does it take place? When exactly does it take place? And what's the importance of this process taking place? Now, the first thing here is what the process is. This is the production of an identical copy of the DNA of a cell. So the same cells that you have in your body. Before cell division, that DNA will make a copy of itself. So think of it like this. If I have a paper that I want to give to everyone, let's say I've got learners in my class, and now I want to make sure that they all receive the same test. I'm going to put this paper under a machine and make copies, and those copies will be identical to the original question paper. Now with DNA, it's not about DNA going under a machine. We're going to show you how exactly DNA is able to make an exact copy of itself. So you can end up then having the original DNA and another DNA in the same cell. That's what the process is. Where exactly does it occur? Where does DNA replicate exactly in a cell? In the nucleus. We spoke about the nucleus. So remember the nucleus, and you will get the DNA in a form of a chromatin network. This is where DNA replication will take place. They will ask you, when does this process take place? It takes place during a phase that is called interphase. So there's different phases during cell cycle. There's interphase, there is prophase, there's metaphase, anaphase, telophase. Those are the phases in mitosis. Then there's other phases in meiosis. So, but let's not stress about the others at the moment. So it happens during this particular phase whereby this DNA will make an exact copy of itself, which is another DNA in the same cell. Now, why is this important? Why does DNA need a replica before cell division? Now, we need the material, which is the DNA itself, the genetic material, to be shared amongst the cells. So if we bring in a little bit of grade 10, cells divide all the time. The reason me and you are grown now is because of our cells have divided. There's different types of cells that we have in our bodies, but that is not important. Now, in a case whereby a cell divides, each and every cell needs to have the genetic material, and under mitosis, it will need to be the same genetic material. Now, when these cells divide, if this cell divides, it's then going to form two cells, which will then be called daughter cells. It will form two cells, which will be called daughter cells. Now, during mitosis, these two cells will need to be identical to each other. And identical is not necessarily about people looking the same and all of that. It's the genetic material, the code itself, that TCA sequence will need to be the same. That's what we mean. Now, this cell over here will need to have the same genetic material as the cell. But in order for this to be possible, we need the original DNA in the parent cell, the cell that is going to divide, to be able to make a copy, meaning another DNA, so that at the end of cell division, this DNA can be shared between the two cells. That's the main goal. So if your screen goes on and off, just know I'm trying to erase. It should come back. It should not go on and off for more than 3 seconds. It should come back. All right, so that's what we are then having here. So that's the main reason that DNA replicates or makes a copy of itself. They do mention it here. It doubles the genetic material so that it can be shared between the resulting daughter cells during cell division. So the question will ask you, state the significance of DNA replication. These are the 2 points that you need to make, or that you need to write down. So one, it doubles the genetic material, and life sciences is so unfair because there's keywords that we need in your answer for you to be given a mark. So doubles the genetic material needs to be there, but now it's not just that. The tick is here at the end. So the reason the genetic material is doubled is because it needs to be shared between the two cells, what has been mentioned there. Now, when that happens, it will result in the formation of identical daughter cells. The cell that divides, we call it a parent cell. Then the resulting cells, we call them daughter cells. Now, due to this DNA replication, each daughter cell will obviously be identical. So the DNA will be the same. Now, then the question would be, how exactly does this process take place? Let's look at what we've covered. We've covered the what. What is this process? This is when DNA makes a copy of itself. Where does it take place? It takes place in the nucleus of the cell. When exactly does it take place? During interphase. Why is it important for this process to take place? To double the genetic material so that it can be shared between the daughter cells during cell division. Okay, so we have all of those 4 points now. We need to know how exactly does it take place. We are going to look at the points. I'm going to skip that because it's not important, so I'm here. Please do make sure that you are also on the slide. This is very important. I'm going to check something. Are you guys still here? I need thumbs up to make sure that you're still here. You're still following. I have quite a lot to cover before I start taking questions. Thank you for those thumbs up, but please feel free to stop me any second and say, ma'am, please do explain this. Now, with this slide, please make a note that you will be asked to describe. There's very high chances that you'll be asked to describe. And when you are asked to describe, it will be for 6 marks. They can ask you to describe. I'm just going back to my notes. Or they can give you a diagram. You should be able to look at a diagram and say, oh, this is DNA replication. Now, make a note, like I said, when they do ask you to describe this diagram, it will be for 6 marks. Now, this is not a type of question that you will need to skip because you do need those 6 marks. Now, this whole process, remember we said it will take place within the nucleus. So everything that I'm going to point out here, you should understand that it's taking place within the nucleus of the cell. So if you have these notes or you can make a note somewhere, please do make notes of what I'm about to say in terms of all the points. So this is how I'm going to teach this. Sorry. I'm going to go through all the points with you from point number 1 and point number 9. And then I'm going to project a clean page whereby I show you how exactly this process takes place. For now, I'm just going to read the points and emphasize what is important. Then I need you on your side to also have a blank page where you will draw this diagram with me so that you can understand it. So that means you would get your 6 marks if we were to be asked to describe, and then you can get your 10, 11 marks if they give you a diagram and then they ask you questions on it. Now, the marking for this is very strict. The order of the points needs to be correct in order for you to get the 6 marks. The keywords that you use need to be there. You can't substitute certain words here because it changes the statement and it sometimes changes the English. So you cannot. So let's look at point number 1. Remember, we said that this process takes place in the nucleus, and the main goal is for DNA to make an exact copy of itself. Now, when we think of DNA in a natural form, it's double helix. Okay, yes, it's double helix. So this is the natural form of DNA. So the first thing that is going to take place during DNA replication, so I'm going to do this to my fingers to show that DNA is double helix, right? 2 strands that unwind, they are twisted together. The first thing that will take place for this process to happen, we need the DNA double helix molecule to unwind, to become untwisted. However, fun fact, if you use the word untwisted or untwist or whatever, you're not going to get a mark. They need the word unwind. So that's the first point. And please listen to this. DNA has to be there. Double helix has to be there. Unwinds has to be there for you to get the mark. Now, if you just say double helix molecule unwinds, wrong. If you say DNA helix unwinds, wrong. If you say DNA double whatever strand, double stranded molecule unwinds, wrong. Those 4 words have to be there for you to get a mark. So let's picture it. Now, DNA is in its natural form, it's helical. Then the first thing that is going to take place is that it needs to unwind. When it unwinds, what you're able to see is my 2 fingers attached together. Now, the reason that my fingers are attached together is because, remember, we've got bases here. So if I were to draw the 2 strands, which we are going to do, so these 2 strands will be bonded together by weak hydrogen bonds. Then in between these 2 strands, we're going to have bases. Let's say we had a T and an A that side, a C and a G, a C and a G. The pen is a little bit slow, but you should be able to see my letters there. So these bases are bonded together, hence my 2 fingers are attached together. The bases in between are bonded by weak hydrogen bonds. Now, we need the bases to be exposed. So the next thing that is going to happen is that the weak hydrogen bonds between the 2 strands are going to break. Once they break, the molecule is going to unzip. Fun fact, that statement has 2 marks already. In other years, they give 2 marks. In other years, they give 1 mark. So the double helix molecule unwinds. The weak hydrogen bonds break. So whatever that is now attaching the bases is not there. If the molecule breaks, that means the bases are no longer attached together. So that means the molecule will unzip. You have that shape of, when you're taking off your jeans, then you unzip, then you have that shape. It's normally like this. The molecule unzips. I'm writing it down. You should be able to see it somewhere here. The pen is slow, but it should write. Okay, so that is point number 2. What do you need to include here? The word weak doesn't have to be there. So I'm putting it in brackets because it doesn't have to be there. But hydrogen bonds between the 2 strands break. There's a tick there sometimes. Then after they break, the molecule unzips. It's not underlined, but this is also important. I cannot help but stress this out. The words unzip and unwinds tend to be confused during exams. the molecule unwinds first. It can't unzip first, then it unwinds. So the order needs to be correct. Also, you can't say the bonds are going to break and then the molecule unwinds. No. If you are baking cookies, I don't know how to bake, but I'm trying to think. You mix the flour and water and milk and eggs first. And then when that is done, that's when you put everything in the oven. So if someone were to ask you the steps that you need to follow when you're baking, you're going to say that. You're not going to say, put everything in the oven, take it out, add milk, add flour. That makes everything wrong. So the order of this is very important. It unwinds first, and then the bonds break, then the molecule unzips. Point number 3. This word is very important. It's not highlighted, but it is very important. Each strand serves as a template to form a new strand. Now, in DNA, we have 2 strands. We have strand 1 and we have strand 2. So after the 2 strands have separated, we are going to use both strands. So if you don't want to use the word each here, please make a note. You can use the word both because we are going to use both strands for them to act as a template. And this will make sense once I do the diagram, which I will do now. So we are going to use bases on both strands in order for us to form new strands, which we'll call complementary strands. Now, how are we going to form these new strands? We are going to use DNA nucleotides. That is important. This process that I'm describing is very similar to a process called transcription, which is the first phase of protein synthesis. If you don't master this process, that is going to confuse you when we do protein synthesis. So I need you to understand this. But now, where are these nucleotides coming from? They are coming from the nucleoplasm. Last week, we spoke about the nucleoplasm. We said the nucleus is made up of a membrane, and then inside that membrane, you have a jelly-like substance, which is a nucleoplasm. Now, these DNA nucleotides are found in the nucleoplasm. Then they will attach themselves to complementary bases, to the bases that are opened here. So we will do it together. I'm just reading the points. Now, when that happens, A will complement T and G will complement C. Now, when we get to this point, we are going to end up having 4 strands instead of 2 because we will have an original strand and a complementary strand. The original strand will be the one that acts as a template to form a complementary strand. Each new molecule will consist of 1 new strand and 1 original strand. At the end, we will have 2 genetically identical DNA molecules. I think it starts being confusing now because we are not seeing the vision. All right, give me a second. I'm about to share a blank screen. It will just be a white screen where we draw. Please also get a paper on your side to also draw all of this. So just give me a second. I am going to show you guys the screen which we'll work with for the process. Then it will make sense. After we do the diagram, it will definitely make sense. Okay, I'm still loading the screen. It's fine. There you go. Okay, so a white document. Yeah, it should be on your side now. I'm coming to check up on you guys. Wait. Okay, thumbs up if you can see the white screen. Thumbs up if you can see the white screen, guys. Let's just give it a few more seconds. It should come up on your side. I'm not seeing any thumbs up yet. Guys, are you still there? I'm not getting any reaction. Are you still there? Guys, are you still there?
The white sheet hasn't appeared as yet, ma'am.
Oh, okay. Wait, let me double check. Okay, it's showing on my side.
Now it's showing on my side as well.
Okay, thank you so much for that response. Let me check on other people. Guys, please do thumbs up if you can see the white screen. It should just be a white empty paper. Okay, I'm not getting any reactions. Let's
Guys,
see.
you must click on the video where ma'am is speaking, and then it's gonna give you an option to click on the other screen. It's showing white.
Okay, thank you for that. So it's not just a matter of it popping on your screen. You have to click somewhere. I'm seeing thumbs up now.
Yes, they must click on the video that you are talking on the live video. Then it's gonna give them an option to click on the white one.
Okay, thank you so much. I got worried there. I was about to send a message because I was not hearing any sound or anything. All right, thank you so much, Leander. All right, guys, please, if you don't have a paper or anything with you, you're not gonna get this. Please have a paper so that we can do this thing together. All right, so this is DNA replication. So my pen is a bit slow, but you should be able to see everything that I'm writing. DNA replication. So when we speak about DNA replication, we are going to use all the points that we've just done together. DNA replication. Something is definitely happening and I'm not sure what. Okay, let's see. DNA. Okay. DNA replication. Oh, I'm really struggling with my screen. Please just give me a second. It's just coming and going. I'm not sure what's happening. I'm not sure if it's this software. Okay. It's fine. Let me see. Okay, yeah, it's fine now. So I need you to draw a circle. It doesn't have to be perfect. That circle represents the nucleus because that's where this whole process is taking place. So the circle represents the nucleus. That is where this whole process is taking place. So please do draw the circle. I'm also drawing my circle this side, but it's not drawing. Oh my gosh. I might have to disconnect this. Yes, I'm listening.
Yeah, The white sheet has been gone since you started drawing. You are aware?
Okay, please just give me a second. Let me log out and then come back because I'm not sure what's happening. I'm just gonna, you don't have to leave. I'm going to leave and then I'll come back. Okay, I'm back. I'm trying to reload everything. This is kind of stressful because now I've tried everything today. It's probably also going to disturb the recording. Let's see. Okay, looks fine. Okay. It should be back on your screen, guys. I'll just give it a few seconds. It's fine on my side. But yeah, I think if it continues to do this, I don't know, because it's frustrating me. I'm seeing a few thumbs up. Okay. All right. Thank you. Let me try again. Um, yeah. Okay. That is DNA replication. So I said we need to draw a circle. There is my circle. It doesn't have to be perfect. It's just a circle representing the nucleus where this whole process is taking place. Now we are going to remind ourselves of the first point. We said the DNA double helix unwinds. So there it is over there. It's going to unwind. When it unwinds, we need the weak hydrogen bonds in between the two strands to break. When those bonds break, the molecule will unzip and then we get two separate strands. That's what we are drawing here. So this is strand 1 and this is strand 2. Thank God the pen is also quicker now. It's also faster. All right. So I need everyone to be awake and get this. So remember, step 1, the molecule unwinds. 2, the bonds break, the molecule unzips. We end up having two separate strands. Our strands are separate because we no longer have bonds that are attaching the bases. I'm going to take out my pens, which I'm going to use as an illustration here. So I've got 2 blue ones and 2 red ones. Okay, so these are the strands that we now have. These are the 2 separate strands. They were once attached to each other. The bonds are broken now. Then we're having 2 separate strands. Remember, once these strands are separated, we are able to see or identify the bases. Please do write the bases with me. So I'm just going to create a sequence of bases over here. And then we are going to follow those bases in order for us to explain this process. So I'm just having a sequence here that is T, C, G, A, T. I'm going to give you a chance to also write it down. I'm happy with my pen now. It's a bit faster. So it's T, C, G, A, T. These are the 2 strands. Now I need someone to unmute and tell me the bases that are on the strand on the right. Now remember, these are bases that were initially paired to these bases that we can see on the strand on the left. Anyone can unmute and tell me?
A,
Yes, I'm
G,
listening. The
A,
first one here.
A,
A, thank you.
G,
G,
D,
C,
T,
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 T.
and A.
Thank you so much. Hey, I didn't even see who was talking. I couldn't go back that side to see who was talking. Thank you, thank you, thank you so much. All right, so we've done the first 3 points. Point number 4 says, okay, I want us to label these strands. Now, point number 4 says that both strands or each strand is going to act as a template. Now, a template is something that we follow or something that we use to make something else, right? Now, that means these 2 strands, this strand over here and this strand over here, will be known as the template strands. We are going to use both of them. So I want you to also label these strands. This one will be the template strand, and this one will also be a template strand. Now, please also add on your side. It is sometimes referred to as a template strand, but sometimes it is referred to as a coding strand. You can even add that here, coding strand. So if they don't call it a template strand, they call it a coding strand. Now, what is this coding or template strand? When we look at the next point, it says that free DNA nucleotides are going to attach themselves into the exposed bases. Where are these DNA nucleotides? They're actually floating in the nucleoplasm. This area here, the whole nuclear membrane is filled up with RNA and DNA nucleotides. But the ones that we are going to use is the DNA nucleotides. How are we going to know which ones are DNA, which ones are RNA? I think it's the base itself and also the sugar. Remember, that is the nucleotide. So all of these are floating in the nucleoplasm. Please make a note. It can't be the cytoplasm. That's a common mistake. It's not the cytoplasm. The cytoplasm is that jelly-like substance of the cell. The nucleoplasm is the one for the nucleus. Now, let's imagine this. This is a strand, and this is another strand. I'm going to start with the left strand first. Now, all of these free DNA nucleotides floating in the cytoplasm, and this is controlled by enzymes. We don't need to know the names of the enzymes and all of that. Now, free DNA nucleotides are going to attach themselves or complement themselves to the exposed bases. Once they do that, a base will come here and another base and another base and another base. Now, these bases are going to be bonded together, which will then form a new strand. I'm going to use a different color, and I will start with my left strand. So what am I basically saying? I'm saying there is a base that is going to pair with the T and a base that is going to pair with the CGAAT. Now, how is this determined inside of the nucleus? It's determined according to a base pair. So the pair that needs to pair with T is the one that will attach here. So an A is going to float and float and float up until it goes, ooh, there is T, there is my partner. Then they bond together. So a new strand is going to be formed here. Now, this strand is going to be formed by bases that complement, the word complement is important, that complement the bases that are exposed. And A will float and float and float, find a T, boom, they are bonded together. They will be our A here. Then another DNA nucleotide floats, a C, and a G, A, T. Okay, there's a G, boom, it finds a C. You get a G here. Then, since we are having a G this side, a base that is going to complement that, that will be a C. Then since we have an A on this template strand, a base that will complement that will be a T. And then here will be a T, and here will be an A. Remember, what do we call these? We call them bases. And where are they from? They are from the nucleoplasm. These are the free DNA nucleotide. But remember, if this is a nucleotide, this is a base. So we are focused on the bases. Same thing happens to the other strand. Remember, you're having the left strand, and now it has a strand that has been formed by the bases that complemented the exposed one. Now, you need to think about it like this. How were we able to form this strand in red? We used the bases in the template strand. The only reason we are having this sequence of bases is because of these bases that are exposed. T is only here because it pairs with A is only here because it pairs with T. G is only here because it pairs with C. So this sequence of bases is determined by the exposed bases in the template strand. Same business occurs on the other side. Free bases, a T will go, ooh, there's my partner, A. Remember, a single ringed molecule pairs with the double ringed molecule. And there won't be a mistake in this because it's controlled by enzymes. So enzymes are going to ensure that, oh, it's normally more like a lock and key model or theory. Because I know in class I did get a question. So what are the chances of a mistake occurring? Yes, we speak about mutation at a later stage, but there's no mutation in what we are talking about. Okay, so then we'll get the bases. So since there's an A here, meaning we're going to get a T, then a C is the one that is going to pair with G, G with C, A with T, A with T, and then T with A. So this is an A. Now I want us to label the strands. The strands that are in red are called complementary strand. So this strand and this one are called complementary strands. Why are they called complementary strands? Because they are having bases that complement the exposed bases in the template strand. So now our goal has been achieved actually. But what do we have? Let's analyze what we have. We started with one molecule which had two strands, the blue ones here. Both these strands acted as a template in order for us to form new strands which are complementary strands. Remember, we said our goal is for DNA to make a copy of itself, and a DNA is a molecule. Now, how many DNA molecules are we able to see in the diagram that we've just drawn? It is actually two DNA molecules. The question would be, where are they, ma'am? This is DNA molecule 1, and this is DNA molecule 2. Remember, we call a DNA molecule a double-stranded. It is made up of two strands. There is the first strand for this. This is DNA molecule 1, and then this is 2. There is the blue strand, which is the template strand, and there is another strand for DNA molecule 1. Same business is happening in molecule DNA molecule 2. There is strand number 1, there is strand number 2. Let's see if the DNA molecules are identical to each other. Remember I said, when we mean identical in terms of DNA, we are talking about the sequence of bases being the same. Let's look at that. If we were to start over here, in this section of DNA molecule 1, we're having a TA. It's a TA this side. So far we're good. But then when we look at the second set of bases, it's CG. It is C and G. Let's look at this one. Strand on the left is C, strand on the right, G. Okay, let's look at the next one. GC. And this one, GC. Have we achieved our goal? Yes. How many DNA molecules do we now have? 2 DNA molecules. But now what we're able to see is that each molecule is made up of a template strand and a complementary strand. It's no longer two template strands. So we had the two template strands, and now we are having two DNA molecules. I've done the very same thing. My red pens in the middle represent the complementary strands. My blue pens on the outside represent the template strands. So we are having two molecules, not two strands. Two molecules over here. Each molecule has a template strand and a complementary strand. That is the end of DNA replication. I'm happy to take questions. I think everything is good now. I feel like things are going well here. I'm happy to take questions. Anyone with a question before I go back to the notes and re-emphasize? Anyone? Anyone? Yes, ma'am. No, ma'am. Can I get clarity on this? Anything? Kamo? Anything? Okay, I think we're good. All right, guys. Please again look at this and see if we have achieved our goal, which is to make two DNA molecules. Okay. If a question were to ask you, how many strands can you see? They're asking you about the strands. You are able to count the strand. This is 1, 2, 3, 4. How many molecules can you see? It is two molecules because each molecule is made up of two strands. I'm going back to the notes. Just give them 5 seconds. The notes for DNA replication should be back on your screen. I'm going to come to you guys so that I can see. I need thumbs up if the notes are back on your screen. Okay, I'm getting 2, 3. All right, perfect. Now, we need to then look at the diagram and then match the points. Or look at the points and then match with the diagram. Now, a DNA double helix molecule unwinds. Then the bonds will break and the molecule unzips. So it unwinds first, break, and then unzip in that order. Then each original strand, what we mean by the original strand is those template ones, because it's the ones that unwind. Each original strand will serve as a template to form a new strand. The tick is at the end. Then free floating DNA nucleotides in the nucleoplasm. Please make a note right across and write cytoplasm. Cytoplasm. Write a note so that you can basically remember that you cannot say cytoplasm in this point. They will attach themselves with complementary bases. So if an A is the exposed one, T will attach itself. If G is the exposed one, C will attach itself. There. There's sometimes a mark for mentioning this and sometimes there's no mark. Okay, for mentioning this part, A to T and G to C. Now let's look at this. The original strand will act as a template to form the new complementary strand. I think that's what we're able to determine. You had the original strand. It will act as a template for us to form a new complementary strand. Then, since you're also having another original strand, which will have a complementary strand attached to it, you are now having two new molecules. Now, in each molecule, you are having the original strand in blue and a new strand, which is the complementary strand. Now, what happens at the end? Since you are now having two molecules, each molecule will twist back. So since you are now having two molecules, each molecule will twist back and form a double helix. Now, this will result in two genetically identical. Oh, you have to highlight this. Two genetically identical DNA molecules. Those words need to be there. Two genetically, if you just say two identical, no mark. Two genetically identical DNA molecules. Please make a note. You cannot write DNA strands. Write across there and write the word strands. You cannot write the word strands here. You cannot say there's two genetically identical strands. That is incorrect. That is very incorrect. This point over here is not important. The process is controlled by enzymes. That is not important. Remember, 6 marks. Let's go through this process. So this is the repetition, but now you are getting diagrams. What are we able to see here? We are able to see this double-stranded molecule. This is after, obviously, the molecule unwinds. So you are able to see the two strands. And in this case, the hydrogen bonds are still there. The bases are bonded together. Then once the bonds start breaking, you are able to see the shape over here showing that the molecule unzips. Then the strands that you have on the outside, this becomes a template strand and this becomes a template strand. Then these are the DNA nucleotides, excuse me, that are going to complement the exposed bases. When that is done, you will have a complementary strand and the template strand, complementary strand and a template strand. At the end, you will have two DNA molecules, which will then twist back, form that double helix, and then wait for cell division to take place. Now, everything else that I'm skipping is something that I've mentioned. Complementary, template strand. This significance, I think we've covered it. All right, this one is for you guys. Let's do it. 5 minutes. I've been talking, hey? 1 hour, 6 minutes. I've been talking. So this one is for you. You also have it on your notes. So I'm not sure what will work if I project this slide or I project the questions. Or which one is better? Should I take a picture and send to the group? But I believe everyone has these notes. So please do, you can unmute and tell me what will work for you. Then I will do that. Please, I need everyone to do the activity. Okay, is it fine the way I've projected it? Knowing that you guys have notes, you can answer yes, yes, no, no.
Yes.
Thank you, thank you. Thank you, Kiamo. 5 minutes. Please do try your best. Please do try your best to answer. I'll give you 2 more minutes. Okay, are we done? Are we done, guys? Thumbs up. Thumbs up if you're done. If you're not done, just say, ma'am, please give me 2 minutes or something so that I know. Guys, you are quiet on me again.
Ma'am, 2 more minutes, please.
Thank you so much, Kamu. Thank you. Please do indicate when you're done. Just say, ma'am, I'm done, so that at least I can keep track of what is happening. I don't want us to waste time. Okay, are you guys done? If you're done, maybe just a thumbs up. If you're done, I'm just gonna look at everyone now. Okay, I'm seeing a few thumbs up. Yes, Bongo Kuhle. Yes, Annelisa. Okay, I am seeing a few thumbs up. Okay, I'll just give everyone 1 more minute. Okay, thank you, thank you. I didn't see reactions from other people. I didn't see, it's not updating me here. Okay, I see Kamu. I've seen quite a few, Sunny. Okay, I think we can start marking. All right, so I'm gonna need you guys to help me out here with marking this activity. Yes, 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 I'm seeing more thumbs up. Okay, let's do it together. Study the diagram below, which shows a section of a DNA molecule during a process taking place in a cell. That's what we are given. Name the process represented in the diagram. Sanelisiwe?
DNA replication.
DNA replication, 1 mark, thank you. Number 2, name the part of the cell cycle when this process occurs. Kamu?
Ma'am, I wrote interface.
Interface is correct, Kamu. That is interface. Name the location in the cell where this process takes place. The location in the cell where this process takes place. Bongo kuhle?
In the nucleus of the cell.
In the nucleus of the cell. Give labels for 1 and 2. 1, there is 1 and 2. They want labels. So those are basically bases there. Okay. Tabo, please do give us your bases for 1 and 2. What did you say? So for Tabo,
I wrote for 1 I wrote G and for 2 I wrote C.
2 I wrote C. 1 I wrote G. Anyone with a different answer? Thank you, Tabo. Anyone with a different answer?
Yes, I wrote.
Let me come this side so that I can see. Mpo, yes, Mpo.
It was G and T. Did I say?
I'm listening, Paul. What is your one?
Quinine and thymine.
So your one is G and your two is T.
Yes.
Okay, thank you. Anyone else with a different answer for 1 and 2? I'm happy to take another option. Anyone else with a different answer?
For 1, I wrote C, and for 2, I wrote G.
Okay, that's Nelly. 1 C, 2 G. All right, let's figure it out. Only one of these options are correct. Let's figure it out. So this is DNA replication. So that means the strand on the outside will be the template strand. Let's remind ourselves of the diagram. The two strands on the outside will be your template strand, and this will be your template strand. Then these two strands here in the middle, this strand that is formed here and this strand that is formed here, these are the complementary strands. So if we were to just focus on the template strands first, there is a C here. So C pairs with G. So this base here in the template strand is a C. That means it's going to pair with base 1. So C pairs with G. So that means 1 is G because this one is attached to the template strand on the outside. That's how we analyze it. So 1 is G. Now then this becomes easy for us. If 1 is G, this strand over here is made up of DNA nucleotides that are going to complement the exposed bases in the template strands. So if 1 is G, G pairs with C, which will be base 2, meaning base 2 is C. Only one option is correct here. So I know exactly what happened here. Nelly, I know what happened here. So you just looked at this and say, okay, you are having C, that means it's a G here, and this will be the same order. You will have a C and a G. I could be wrong, but tell me if that's how you analyzed it.
Yes.
Nelly?
Yes, and I wrote
Yeah.
it like that.
Okay, so you have to take it from the outside to look, okay, template strand and another template strand, their original bases were actually paired. So this C over here was paired with this one, so it has to be a G. Now, I'm not sure what happened, Paul, please do unmute. You got G correct as the base, but then 2, you got T. Okay, please let me know how you got T.
Oh, Mel, I made a mistake there.
You made a mistake.
Yes.
Okay. All right. Thank you so
Uh,
much.
I
Yes,
made
I'm
a mistake.
listening.
No, I made a mistake there because I know that with the bases, always what you call G
G.
and C paired together, and then A and T paired together.
Okay. Okay, cool. I'm happy to hear that you made a mistake because if you make a mistake in the lessons, that means in an exam you won't make a mistake. All right. Let's do this one quickly. Let's do this one. I'm just going to stand up. I forgot to close my sliding door. So all of these inserts are in here now. So I'm giving you 5 minutes to just do this activity that I've shown on your screen. I'm going to stand up. I'll be gone for like a minute to just go and close my sliding door.
you. Right,
I'm back. You know I'm back. I'm back. You know it. Oh no, it doesn't say I'm back, but yeah. I hope you guys can see me because I can't see myself. Let's see. Okay, there I am. 3 minutes.
Ma'am,
Yes.
I was very much interested. I think you forgot question number 5 on the previous activity.
This teacher. Sorry, state 2 ways in which the structure of RNA differs from the DNA structure. Okay, I think let's allow everyone to finish the activity. Thank you so much for that. I didn't even see. Then, before we start with the second activity, I'm going to do that question. Thank you so much. How many more minutes do you guys still need? Tell me. Do you need 2 more minutes? 1 more? Are we done? 2 more minutes. Okay,
Yes,
let's
ma'am.
see. Okay, thank you. Okay. Okay, let me not rush everyone. Maybe another 2 minutes. And please, you have to get full marks here. This one, DNA replication, full marks. Full marks. Okay, thumbs up when you're done. Ooh, these mosquitoes are eating me. Thumbs up when you're done. Thumbs up when you're done. Can we mark or do we need few more minutes? Can we mark? Yes, no, no, yes, no, yes, no, yes.
Yes, we can mark.
Just 1 more minute, ma'am.
Okay. Okay. Five more seconds. 5, 4, 3, 2, 1. Thank you. All right. I hope everyone is done. I'm not sure what's happening with my screen. It's lagging, but let's hope you guys can see or hear me on your side. Okay, let's start. Study the diagram below and answer the questions that follow. Okay, so we need to be able to study this diagram. What is it? It's a DNA molecule. What is happening here? And all of that. Name the process illustrated in the diagram above. Kamu?
DNA replication.
Thank you. Oh, okay. I almost forgot again. All right. This question that we skipped, guys, question 5, state 2 ways in which the structure of RNA differs from the DNA structure shown in the diagram. So you are going to tell us that RNA, you are basically differentiating. RNA will have uracil, obviously, will have uracil, and you write a whole statement. You don't write what I'm writing. So RNA will have uracil and DNA will obviously have thymine. That's how they would differ, right? So since it's a DNA molecule, it will have a T, and an RNA molecule will have a U. Then you can also speak about the strands. They've said the one that is shown above. So RNA is single-stranded. So if you wrote something about an RNA being single-stranded, that is correct. And then a DNA molecule being double-stranded, that is correct. I can't think of, well, my pen is acting up again. I can't think of any other correct answer. If you mention the sugar, they might argue that you can't really see that because they're saying from the DNA structure shown in the diagram. So in this particular question, you cannot mention that DNA has a deoxyribose sugar and RNA has a ribose sugar because that's not what we can see from the diagram. That's what me and you know. So what we can see from the diagram is that there is a T somewhere. So if there's a T, that means the DNA, and we can also see that it's double-stranded. So those are the answers. For RNA has U, Russell, whereas DNA has T. That's one mark at the end because you're comparing. Then with RNA, it's single-stranded, DNA double-stranded. So that is that. Okay, then let's go back to this one. State the significance of the process mentioned in question 1. So state the significance of DNA replication. Let me see, there's more people on the other page. I'm going to another page. Annalisa?
It doubles the genetic material so it can share between the resulting total cells during cell division.
Tick, one tick. That whole sentence, exactly how it is, one tick. You don't mess up anything. You don't multiply, subtract, 1 mark. So you can be lucky sometimes. They can allocate 2 marks for it. So thank you so much. Identify the parts labeled 1, 2, 3, and 4. There is 1 over here. There is 2. There is 3. And there is 4. Let's see who's going to give us all of these labels. Nati? Nati, please do answer label 1 to 4 for us. Okay, I'm going to move. Nomasondo, please. Nomasonto? Okay. Anyone else, guys? Please do unmute and answer. Anyone?
Hi, ma'am.
I'm listening.
One is phosphate.
One
And-
is phosphate. I want us to look at it again. Is one phosphate? Let's look at it again.
No, sorry, ma'am. Sorry.
No,
Yes, I'm
One-
listening.
One is deoxyribonucleic acid, sugar, I mean.
Thank you. Deoxyribose sugar, not ribonucleic sugar. Deoxyribose sugar. So we've answered one. Then anyone to give me 2?
Ma'am,
Yes.
ma'am, what if you wrote pentose sugar? Do you still get a mark, or is it wrong?
That would be wrong.
Okay.
Do you know why it's wrong? Because they will say, you can see that this is a DNA molecule. So we want the specific name of the sugar that we find in DNA nucleotides.
No, oh, okay then. Okay.
Yes. So if it was an RNA molecule, you would have to specify it's a ribose sugar. If you just write the generic name, it's a pentose sugar, you won't get a mark. So you need to write the specific type of sugar for that specific molecule. For our diagram, it's a DNA molecule. So the sugar we find in DNA is deoxyribose sugar.
Okay, ma'am.
Okay, thank you.
and I think
Yes, I'm
ma'am,
listening.
I think she would have to write that pentose sugar when they say perhaps draw a nucleotide and then they don't specify a nucleotide for RNA or DNA, then she would be able to say that.
Yeah, I think in a generic setup, yes, that would be fine. But even if they were to ask learners to draw, they wouldn't ask a generic answer like that. They will be specific to say, draw an RNA nucleotide. So that should mean ribose sugar, or draw a DNA nucleotide. That would mean deoxyribose sugar. So they would be specific even about the base that you have to include. Okay, but in a generic way, that is correct. But when it comes to questions, you have to be specific. Okay, so then 2, what is our 2?
Phosphate group.
Phosphate group. If you just wrote phosphate, that is correct. The word group is not important. So if you just wrote the word phosphate, that is fine. Then 3. Anyone to give us the answer for 3?
Nucleotide.
Nucleotide. Perfect. Nucleotide. Then 4.
Nitrogenous base.
Nitrogenous base. Please be specific. Which one? Use the guidance from this side to tell me which one. Use these bases to tell me which one that will be.
Diamond.
Diamond.
Thank you. It will be a T. It will be a T. Why is it a T? We are guided by the bases that we can see in our template strand. If you can't figure out which one this pairs with, since they are a bit far, you can start from here. Base 1, base 2, base 3. Okay? Then base 1, base 2, base 3. So this base 3 will pair with this base 3. A pairs with T. Okay? So if you just said base or nitrogenous base, no mark for it. Okay, describe how this process takes place. Please check the marker location there, a whole 6 marks. Yes, Leander?
Ma'am, I wanted to ask from 3, ne? Yes, it
Yes.
is a nucleotide. It is a nucleotide, but is it not important to tell that nucleotide? For instance, if I would say that it is a nucleotide that is free floating, is it not important to mention, or we just say nucleotide, that's it?
For this one, just saying nucleotide is enough, because we are just mentioning that, oh, okay, we are able to identify the sugar which is bonded to the phosphate, which is bonded to a base. So if you mention free floating, we're not going to penalize you for it. We'll still give you a mark. Free floating nucleotide, we'll still give you a mark. But that
Nucleotide.
free floating word is not important.
Okay, for number 4 as well, yes, it is a nitrogenous base, but we don't have to mention that is the complementary one.
No. Remember, when you use the word complementary, we normally use it for the strand. So it is a complementary strand. These bases are in a complementary strand. But if you actually look at this base over here, it's not in a complementary strand. It's in the original strand. There's the original strand over here. So this base is attached to it. So here it's just a matter of identifying what base it is. So when we use the word complementary, that's the strand that is formed. Yes, those bases, I guess one can argue that it's still a complementary base because it's in a complementary strand. But I think for this particular part of the question, yes, I would take that argument. But for this part of the question, we just wanted you to identify the base. So if you say a complementary base, you still have not answered the question. Give us the specific name of that complementary base. So we kind of have to be this specific for this one. Are you answering number 4 for us? This one, the 6 marks one, Leander?
Should I?
Yes, please.
Okay, they say describe how this process takes place. First of all, we need to know that the DNA replication is taking place in the nucleus. So the first step is that the DNA double helix unwinds within that nucleus. And then number 2, the weak hydrogen bonds between the 2 strands, they break, and then they unzip. And then number 3, each original strand serves as a template so that it can form a new strand. And then number 4, those free floating DNA nucleotides in the nucleoplasm attach with complementary nitrogen bases, which means adenine is always with thymine and guanine is always with cytosine. And then number 5, the original strands act as a template to form the new complementary strand. And then number 6, each new molecule now consists of 1 new strand and 1 original strand. And then each new molecule now twists back, which means they wind again back up to form a double helix. And then the result is 2 genetically identical DNA molecules. And then they say each DNA molecule is identical to each other and the parent molecule. Yeah,
6 marks.
Basically that's that.
That is 6 marks. So how we mark, as much as there's 9 or 8 points there, we will look for 6 marks. So don't just say you're going to write the first 6 points, because they can change the mark allocation for that particular year. So be safe and write everything. So thank you for that. So that's how you answer this, guys. I forgot to mention one thing. If you number your points, they can penalize you. You can end up losing that 6 marks. What do I mean? So if you write point number 1, just like you have them on the slides, we have them in numbers so that learners can remember this one comes first and this is the second one. When you write in a test or an exam, don't say point number 1, DNA double helix molecule unwinds. Point number 2, the weak hydrogen bonds break at the molecule and zips. Point number 3, you can be penalized. Life sciences is unfair. You can't get a 0 because you numbered your points. Do not number your points. If you want to write them in a point form, it's allowed, but please do write dashes or dots. Please make a note of this. Write dashes and then write DNA double helix molecule unwinds. Whatever that you want to write in a point form in life sciences, do not number it. If it's a describe or explain kind of a question, do not number. You will be penalized. Last question. Give one location of extra. I'm hearing someone. Is there a question? I'm hearing someone. Come away speaking.
No, ma'am, it
No.
was by mistake.
Okay, thank you. Give one location of extra nuclear DNA. Where can we get extra nuclear DNA? Anyone wants to answer this one? I'm listening.
ma'am.
Anyone? Annelisa?
It's mitochondrial nuclear or DNA.
Yeah, I'll take that. I'll take that extra nuclear DNA. I'll take that DNA of the nucleus. I'll take that. Yeah, I will take that. Okay. Let's see what else we have left. Protein synthesis. We're not going to be able to cover this. We're not going to be able to cover this. So let me project one more activity for DNA replication and then we call it a night. Already it's past 8:30. But yeah, let me do one more activity and then we call it a night. Let's see if I can get it quickly. I wanted to say something. That thing about the points, please, please, please make a note on your notes. That's why you barely find learners getting 100% in life sciences. I'm not sure why the people at the marking center apply all of these strict rules, but yeah, they are there. We know them. Okay. Maybe I might not survive this one. Let's see. Now we can do this. We can do this. We can do it. We can do it. 4 minutes. 4 minutes. This one, 4 minutes. Just even think about it. Just think about it. It's just 4 marks. You are not discussing anything. Just look at it. Just look at it. Just look at it. Just look at it. And then 4 minutes, guys. Nothing extra. You should be mastering this now. It's just 1 mark answers. That's it. You guys are too quiet. Are you there? I'm not hearing anything. No sound, nothing. Are you there? Please.
Yeah, ma'am.
Hey, this online business is so tricky because I'm thinking, what if these people are gone? What if they got disconnected or something? Because it's silence this side. Okay, I'm happy you guys are still here. But we can start marking. Yes? Yes? No? Yes?
No,
Oh,
ma'am.
that's a no. No. Okay. Thumbs up if you're done. This is 6 marks. Thumbs up. Okay, quite a few. A few. Okay, I'm seeing quite a few. Thumbs up if you're done, please, guys. Okay, let's mark, guys. This one is easy. I will even give you the answers. Let's see. The diagram below represents a DNA molecule undergoing an important biological process. Identify the process shown. We are able to see on the outside here. What is happening? Okay. Give me a second. It's going to come back. Okay, yeah, we are able to see the template strand here, a complementary strand, So the process shown in the diagram is DNA replication. The chemical bond labeled 1. This chemical bond here is hydrogen bond. Remember, whether you include the word weak or not, it doesn't matter. Then 1.4.2, give the collective name for the parts labeled 2, 3, and 4. There is 2, 3, and 4. The collective name is nucleotide. Give the full name of the nitrogen base labeled 5. They want a full name, guys. Where is 5 now? Oh, this thing is moving up and down too quickly. Where is 5? There is 5 over here. So that will be thymine. And they wanted a full name. So if you wrote T, wrong, because they've asked you for a full name for base number 5. There is number 5 here. That is thymine. Then, during which phase of the cell cycle does this process take place? Interphase. Where in the cell does this process occur? Nucleus. That's it. That is it with the activities. I think when it comes to DNA replication, you should be covered now. I will send activities and I should be able to send them now because I'll be on my laptop for another 30 minutes. I will send the activities. I'm not sure if my camera is off. I will send the activities just now before we sleep. These activities include DNA replication questions and protein synthesis questions. Please note that we have not done protein synthesis. We'll do it next week. In terms of covering content, all of it, it's not going to be possible, but If you feel like you can answer protein synthesis questions, I would really, really appreciate that, so that when we do the lesson next week, I touch up on protein synthesis and then we mark. But let's see how it goes. All right, I'm ready to call it a night. But before that, I'm happy to take questions, comments, suggestions, anything that you want to say. Anything
Ma'am?
at all. Yes, I'm listening.
Yeah, I remember we came across these two last questions, ne, on our first activity. And then how they asked the question on the first activity, they say, name the part of the cell cycle when this process occurs. Ne? Now here they are saying during which phase of the cell cycle does this process take place? Ne? I wanted to ask, when we talk about interphase, we're talking about it's a process, most it's a phase.
It's a phase, yes.
Yeah, but why would they ask that question that way on the first activity, where they are saying name the part of the cell cycle? Do you get my question?
So yeah, I do get your question. So the cell cycle can be regarded as having different parts. So they will be, or different phases. They can say a phase or a part. So they're just using different words to try and confuse you, but you should know your content. If you know your content, you won't be confused. Someone can argue that it's not really part of the cell cycle, it's a phase, but if they phrase it like that and then you understand what they mean, you just need to answer, because you have the interphase, you have the growth phase, and then you have the other phases, which is prophase, anaphase, telophase, and all of that, metaphase. So the correct terminology would be to refer to it as a phase, but if they've referred to it as a part, we'll just have to use our content knowledge to answer. So I think it's a good question for you to question it, to say, why is it called the part this side and the phase the other side? But I think the guidance there is cell cycle. Once they ask you about a part in a cell cycle, a phase in a cell cycle, I think interphase should come to mind. But I've never looked at it like that, but you are correct.
Yeah, because it felt like they're asking literally, or something that you
part.
Yeah, a part. Okay, thank you, ma'am. Thank you.
Okay, thank you. Anyone else, guys, before we say bye to each other? And remember, we don't just leave the meeting. So anyone else? Okay, I think we're good to go. Ooh, close to 2 hours. All right, I am going to pray and hope that the whole lesson was recorded. And I will only know that tomorrow morning, because Teams will only send me a video is ready tomorrow morning around 5 o'clock. Then I will let you know, guys, on the group. But yeah, thank you so much for today. And you know what, just being patient with my pen and my pad. This is brand new and it's supposed to work way faster. I'm able to see everything here, whatever that's on my laptop. It's supposed to work way faster and it's so expensive. So I'm not sure what is happening again. I'm just going to try and seek IT assistance. Maybe there's just something that I'm not doing right. So that's why we're having all of these mishaps. But thank you so much for taking the time to join the lessons. Thank you so much for being patient with me as I'm trying to figure out these things. But yeah. All right. Thank you. Have a good night, guys. Remember, you need to unmute and say bye. Then you can bounce. I'll see you next week.
Bye, ma'am.
Bye.
Bye.
Bye, guys.
Bye.
Bye. Bye. Bye. Bye. Bye. Bye.
Bye.
Bye. Bye, guys. Tot ziens. Tot ziens. Goeienaand.